Great, great. Okay, yeah, let's start. So, hi everyone. I'm Vincent. I'm a postdoc in the Dorenstein lab, and today I'm super excited to give this workshop about reverse metabolomics with Victoria.
Hi everyone, my name is Victoria. I'm a staff researcher in the Dorstein Lab. Super excited to be with you all here today, and thank you for coming.
Nice. I think now we can start with the science. Oh, let me start introduce the team first. So we have Elena and then William and then Shibe that will be online to answer your question in the chat. So if you have question, just feel free to ask them in the chat during the presentation. Um, it's our first ever workshop on reverse metabolomics. So if there are some challenges or hiccup along the way, we are going to try to address them. It's possible. So usually we don't give that workshop to that many people here, and that people are still joining. So if there's any problem, yeah, just feel free to reach in the chat. So let's start here. So I will assume that most of you and us included works with molecules or feature. It could be a molecule that you know, a feature that is unknown that you just recently discovered. It could be a feature that you think it's important, or you try to explain the biology or try to find its function. And you might want to know if these molecules are found in the human body, so or in different organs. So if these molecules are found in liver, in the gallbladder, or in the small intestine. You might also be interested to know what is the distribution profile of these molecules. Are these molecules found in mice? Are they found in human or other organism? And we also think that reverse metabolomics can be leveraged to answer if is mice the right model or the right organism, which are the organism that could be used to study your specific molecule of interest. You can also have different, you might be interested to know if your feature of interest or your metabolite is associated with a specific diseases. So is this molecule more prevalent in Alzheimer's patient, or is it more with virus infection like COVID-19? So you might want to also add other information to this. So let's say you found a feature that it's found in the gallbladder or in the liver. And now you want to know the origin of this molecule. Is this molecule associated with a specific microbes? Is it plant derived or is it food derived? So you might want to know if this molecule is coming from the diet. These are the type of question that reverse metabolomics can help you answer. So for today's workshop, our goal is really to provide you with sufficient information to get you started. So we want to provide you all the basic to do reverse metabolomics on your own. So what is coming is we have a step-by-step guide that is accepted in principle in Nature Protocol to guide you step by step to do reverse metabolomics. So it's super descriptive. You can follow this protocol step by step. It will be available in the near future, and we are planning to also put a preprint online in the coming days and weeks. So what is reverse metabolomics? So reverse metabolomics is a data science strategy that would take the MSMS first approach to search and probe public data to attach any biological information that you can find. So right now we leverage reverse metabolomics in 3 different papers that have been recently published, and one is in a preprint right now. So we mine public data for bile acid and nasal lipids. If you haven't read those papers, I encourage you to do so. They're super descriptive, and then we also explain a lot of biology behind these papers. And then for today's workshop, we're going to be focusing on 3 small molecules. So phenylalanine conjugated to cholic acid, histidine conjugated to a C4, short-chain fatty acid, and phenylalanine conjugated to C4.
Any
So,
questions so far?
uh,
Anybody? If not, you can go on.
right.
I like to give opportunity to ask questions. So
Yeah, no, that's great. Thanks, Peter. And then in reverse metabolomics compared to traditional, so traditional untargeted metabolomics usually start with a question. So you have a question, you plan your experiment, you do the sample collection, and then you generate the data. But in reverse metabolomics, usually start with a MSMS spectrum. It could be a molecule that you know the structure, or a molecule that it's unknown but seems interesting to you. And then you use the mass search tool to search the public data. And then we conduct a metadata-driven analysis at the repository scale level. So this is not limited to a single study or a data set. But here and then here, by attaching all this information, then you can formulate your hypothesis or your research question. So there's two ways that we recently leveraged to acquire an MSMS spectrum and then attach information. So the first approach was the synthesis-driven reverse metabolomics approach. So here we did hypothesize or predicted that some bile acid or some molecule might exist in the biology. So we synthesized them and then acquired their MSMS spectrum. The other approach that we use was the MassQL tool that we just use in our cell publication. So here, MassQL is a universal language that allows you to find patterns in the MSMS spectrum. So here, we will queries to target some bile acid dianistic fragment ion, the pattern of bile acid, to search in the public domain and collect identical or similar MSMS spectrum for the pattern that we just described here. So there's multiple ways one can obtain an MSMS spectrum. It could be in the supplementary materials of an article. It could be on one's computer. There's multiple ways that you can obtain an MSMS spectrum or in the different library or different databases. And we also have, besides mass, we also have our domain-specific mass that try to answer the origin of these molecules, where these molecules are coming from. So you might want to know if your molecules are microbial derived, if they're plant derived, or food derived. These domain-specific mass have their own curated metadata, and they have their own interface that you can just search if your molecule is found in these databases. And reverse metabolomics heavily depends on metadata. So we conduct a metadata-driven analysis, and now it's possible to search against all three main metabolomics repositories. So we have GNPS Massive, Metabolites, Metabolomic Workbench. All this data has been harmonized and indexed, and you can also, if you're interested in knowing more, you can just see the preprint that it's available on ChemArchive. And here it's really important to, I want to emphasize the importance of uploading your metadata and a comprehensive metadata with your data deposition. Using Redo, it had never been that easy to create your metadata with a curated, created ontology and vocabularies, a control vocabularies. And this will have a huge impact either for your own research, but also for your community. Because reverse, the more data and the more metadata we have, the more powerful reverse metabolomics will be for the community and for your own research. So here, this is our new interface to upload your metadata. This is now available using the following link here. And then you can click on the contribute your data, and here you can upload your data. It's pretty simple. It's a nice workflow. Now, what we have available, it's over 600,000 files, over 3,000 unique data set. And we're covering a lot of different taxonomies, some bacteria, mammals, plants, primates, rodents. We have a lot of human and mice data, different body parts, and different diseases. So now here you can just explore this dashboard and then the documentation to know how you can contribute your data to the new Pan Redo.
Thank you, Vincent, for the great introduction to reverse metabolomics. I want to go ahead and invite any questions anyone has. I see one in the chat. MSMS pattern depends on instrument and collision energy. What's the default setting we should use to collect MSMS? So I would say in metabolomics, there is no default setting. Everyone has different instrumentation, different availability. And thank you, Helena, for answering in the chat. And one of the beauties of reverse metabolomics is you can start with any MSMS and search the public domain, and it will change the hits that you get depending on what your input is. But really, I would say it's kind of a one-size-fits-all workflow.
So, but I think there's still some recommendations that could be provided just on the MSMS alone. If you go and collect data, there are some default settings, and I don't know what instrument is being used, but if it's, let's say, an Orbitrap-based system, typically whatever the 30 value is, typically it's set to 30. And that is not really a meaningful value, but it's consistently what people often use on Orbitrap system. If you have a TOF system, it's not unusual to scan multiple collision energies and then combine them afterwards. One of the things that we do when these searches are happening, we essentially apply a square root. So we decrease the high intensity ions, increase the low intensity ions to sort of overcome some of those instrument differences. But in the end, we rely on just having access to so much data that you will start to be able to begin to interpret biology. So you may miss some because of your particular instrument settings. But there should be enough data in there that should be overcome. The alternative is broad settings for MSMS searches, but that comes at a disadvantage with more false matches.
Okay, thank you, Peter.
Thanks, Peter, for the awesome answer. Does anyone else have any questions before we move on to start the hands-on training? All right, okay, let's get into it. So firstly, obtaining the MSMS spectra. So today we want to showcase our public GNPS library, where there's hundreds of thousands of available spectra to choose from. And that's where we're going to be getting ours today. All of you guys also have the ability to input reference spectra into this library. And if you're curious on how to do this, I please invite you to check out the GNPS documentation. Next, we want to show you our masked query tool, which is also publicly available in GNPS. This is the computational workflow that takes our input MSMS spectrum and searches it across all three repositories, GNPS Massive, Workbench, and Metabolites. We're going to show you how to associate the metadata that's available on PanRedo, which we talked about earlier. And really, this is the juice of the hypothesis-driven analysis. So from this combination of data, you're going to have a lot of options for pan-repository analysis. The last step in reverse metabolomics, we call validation. So this is going to be experimentally deciding and validating these pan-repository analyses. So this is experimentally driven. We're not going to be able to show you that today. We can definitely answer questions and give you an introduction into how to do this. But today we're going to get you to the hypothesis stage. One of the key things that brings the GNPS framework together is called a USI, a Universal Spectrum Identifier, which is basically a URL for each individual mass spectrum. And these, again, are the three small molecules that we'll be using today and the USIs for each of them. So let's begin by going to the GNPS library. Vincent, if you could post the link in the chat for everyone to click on if you're following along for the hands-on. And we'll give a few minutes for that. Thank you, Helena, for putting in the chat how to add your spectrum in the GNPS libraries and also how to make your data public in GNPS Massive. We have documentation for all of these steps. And if anyone has any questions while we wait for everyone to log in, I'm happy to take them. All right. Maybe we've all made it to the public GNPS library. If anyone's having problems with this step, please let us know. So that first small molecule that we're going to be searching for today as an example is phenylalanine cholic acid. So under the compound filter, go ahead and type in PHE -CA. Then you're going to get a list of all of the phenylalanine conjugated acrylic acid that's available in the GNPS library. Something that's unique about the GNPS libraries is that we allow multiple spectra from different people, different instruments into our libraries, which of course advances the amount of mass searches we can do using this approach. And I'll be giving you guys time to kind of play around with the interface. Maybe you've never used GNPS before, but if you do scroll down, you're able to see the spectrum. And I invite you all to go to page 2 and click on the last spectrum for the one that we'll actually be using today, uploaded by Emily Gentry. As you can see, it shows you the USI above the spectrum. If you click that, it'll take you to our metabolomics spectrum identifier resolver. This is where you can visualize the two-table MZ intensity as well as the spectrum for any USI in the GNPS framework. And Vincent just sent the link for that as well. And maybe I'll give a few seconds to kind of play around with these 2 pages if anyone has questions.
Hi, I have a question. So when we click on each of the peak, it gives us another page. So what does that page show? It's information that it gives us.
When you click on a different peak, it takes you to a different page. Do you mean on the GNPS library or on the metabolomics resolver?
So
I
on
think
the, yes,
I
on the
think it's opening
USI.
up the metabolomics resolver that leads you to the axial spectrum itself that you can download, or there's also you can grab the USI from there. You can label the peaks. You can then create a snapshot of it and put in papers and such.
Oh, I see. Yeah, it's the same spectrum, but with labels and stuff, right?
Yeah, so you can choose your labels and
Uh,
specific how you want to look at view it.
yeah.
Yeah.
Okay. Thanks.
Yeah, so there's a lot to play around with on the USI resolver. Yes, thank you, Helena. It will show all of the fragments. Okay, maybe I'll move on. All right, the next tool in GNPS that we're all going to be going to is the FastMast. We call it FastMast because it's going to search the spectrum across all 3 repositories within seconds. So Vincent just put that link in the Zoom chat as well.
Yeah, so this is all LC-MS data in metabolites, metabolomic workbench, and GNPS massive. And so all these spectra are indexed, making searches reasonably fast. Currently, we have over 2 billion MS-MS spectra indexed in this infrastructure.
Thank you, Peter. I'll give everyone a few more seconds to head over there. And Vincent, maybe you can post the USI for the phenylalanine cholic acid in the Zoom chat. Yes, right now we only have LCMS data. No GC as far as I'm aware of. for the mass search.
Correct. No GCMS, because that requires deconvolution unless the GCMS is done, let's say, a modern Orbitrap system where you also have a precursor mass and then the data structure is the same as LCMS. I did recently learn there's some other group, and I don't remember where I saw the paper, but they're considering of creating a resource for GCMS like this.
Yeah, that would be awesome. I mean, expanding these kinds of tools would
Yeah.
certainly help the whole community. So I'll invite everyone to start with the phenylalanine cholic acid and paste that in the spectrum USI section of the FAST search. And we will be using the default parameters today. So searching against pan-repo means pan-repository. So searching again across GNPS Massive, Metabolomics Workbench, and Metabolites. Then click on search. And maybe we'll give a few seconds for that to load. And if anyone has problems at this step, please let us know. And we do invite you to look around on this page. You might see some tables showing the delta masses of all the different matches. But most importantly, you're going to get a table of all of the matches for this compound in the public data. No results yet for me. Yeah, I'll wait a few seconds. Sometimes this step, depending on which computer you have. And of course, there's many of us probably trying to use this tool. So we'll give a few more minutes. Thank you, Celeste. Which library to use? Maybe we can go back one page.
So the library that you should use is the Pen Redo 2024 1112, which is the last one. Every time we do upload this library multiple times per year. So just look at the date, and it should be the last one that you should be using if you want to search across all three untargeted metabolomics repositories. So I encourage you to usually always select that one if you want to search across all three of them.
Yeah, good point, Celeste. Thank you. So the USI starts with the mzspec. If anyone might be having problems with this step, maybe that's the reason.
Thanks, Helena.
Maybe just one more minute.
I mean, it should be mostly done for most people.
Mhm.
Is it not done for some people? If they can let us know.
Okay, I guess we can go ahead and move on then.
Yep.
So again, if you scroll down, the first thing that you'll see is this table with all of the mass searches. So if you take a look, we can look at this table together for a bit. So it shows the USI of the match in the public untargeted metabolomics data, the charge of the molecule, the cosine similarity score. For those of you who don't know, cosine of 1 means it's like a perfect match, and it shows that there's 14 matching peaks, and it's found in this data set, 91795. This means that it's a GNPS massive data set. So if you head over to massive.ucsd.edu, which we won't do today, you can basically download all of these files publicly of that data set. So the first thing that we will do is hit that export button at the top. And wherever your downloads go, maybe that's the downloads folder, you can go ahead and rename that file to the name of the compound, in this case, phenylalanine cholic acid. Thank you, Shippei. Yes, so we did send an email out yesterday with a folder for everything that you'll need today. Did anyone not get that email? We did ask that you put that folder in your desktop. Okay. That is fine. No worries. Yeah, no worries. We can also do it manually. For those of you who did get the email and you have the folder, just hang tight with us. So we recommend putting a folder in your desktop named CMMC Workshop. And when you export this file, it will be named data.csv. You'll rename it to phenylalanine cholic acid.csv. And we recommend to put that file in your folder directory. Thank you, William. Yeah, and there's the zip folder from William in the chat. All right, so you're welcome to repeat the fast search for those other two molecules, phenylalanine C4 and histidine C4, these two N-acyllipids. But the important thing is just to have the data tables in your folder. So does anyone not have the file that we either just sent in the Zoom chat or in the email yesterday? That's awesome. Okay, I think we've had enough time to kind of play around with this interface, and maybe we have all our data tables ready. We also did recommend to install RStudio, as that is the language we'll be using for our script. Does anyone not have RStudio or still needs to download this? Okay, I think that means that everyone has RStudio. So go ahead and open that up. So we do have an updated version of the script, which we included in the zip folder. But for regular updates on it, since the tools are constantly improving, it will be on Vincent's GitHub. Vincent, you're welcome to add the link into the Zoom chat. And it's always available to download here. And if anyone needs to do so, we did include it in the zip file. Thank you, Vincent. All right. So let's go ahead and open up that .r file in our RStudio. I'll give a few seconds for that.
Yeah, apparently the R script might not be in the zip file for some reason. So just go back maybe one or two slides, Victoria, just to showcase how to get it. Good, here on the GitHub. So if you just use that link here, so it should lead you to that
Maybe
interface.
provide the link in the chat as well.
Yeah, it's there. It's there. It's also in the chat. And Shipe and Victoria just send also the script here that you can just download and then open in your RStudio interface. So now just to recap, so in the chat, William just posted the folder that contains all the tables that you're going to need for today's workshop on the R environment. And then you have access to you have the script that was provided by Shipe and Victoria here. And in the folder that you just downloaded, there will be the redo metadata. That is the TSV file called all sample information. And inside that folder, you're gonna have the input fast search file. So the one that we just did together for histidine C4, flanellin C4, and the flanellin cholic acid. And this will be used for our directory in the RStudio that we're gonna walk you through it in a second.
Thank you, Vincent. All right. Seems like everyone is caught up, but please let us know if you have any questions or need some help. The first thing that we want to do once the script is open is to set our working directory to that folder that we just provided to you guys called CMMC Workshop. You can do this by hitting Session at the top, Set Working Directory, and choose Directory. And then a pop-up of all of your files will open, and just choose that CMMC Workshop folder. Okay. So the three packages that we'll be using today as part of our script is the data table, tidyverse, and pheatmap packages. If you have these already installed in your environment, then no need to re-download them. But if you've never used R before, or you know that you don't have these packages, go ahead and uncheck or unhashtag those lines of code. For anyone who maybe hasn't used R in the past, you can hit either Control-Enter or Command-Enter or the little Run button at the top of the R Studio. And if anyone has any questions like that, feel free to ask. And I'll give a few seconds, and sometimes the packages can take a minute to load. And then we can go ahead and run the library for all the packages to load them into our environment. So the library data table, library tidyverse, and the library pheatmap, go ahead and run those lines as well once the packages have been installed. All right, assuming that everyone has gotten to this step, we do want to specify the folder path, which is the input fast inside of the CMMC workshop folder, which was our working directory. So just to recap again, these are the data CSV files from the fast search. Okay. Next, we want to import the all sample information.tsv, which is the pan redo metadata that we already went ahead and downloaded for you. But again, you can find that on our pan redo dashboard that we showcased earlier. And we'll use this function to import it. So I'll assume that everyone has run those lines of code. Just to make sure that the metadata was imported. On the right side in your environment, if you click on that data frame, it should pop open and show you what the table looks like. And you're welcome to take a look. Moving on. Now we want to get all of our CSV files from the FastMass search and collect them and merge them into one table. And add a filtering step for the delta mass. We only want to keep matches that are within 0.05 mz. And if you want to go ahead and open that molecule's interest filtered data frame, you'll see that each mass search now has the corresponding compound name for that mass search. And all three molecules should be in one table. So the phenylalanine cholic acid, the histidine C4, and the phenylalanine C4. And please let us know if you're seeing issues in these tables. And basically what this table is saying is that histidine C4, for example, is found in this scan of this data set with a cosine of, for example, 1. Okay. Moving on.
Nice, thanks, Victoria. Yeah, let's just go back one more slide here for 1 second. So the way we designed this script is trying to make it as easy as possible. So we just aim to specify 2 different directory here, which is your directory where you have all your files, and then a second directory where it points to the CSV file. And from there, everything could be run automatically. So we're gonna go to the code, explain it, what it does, but we're trying to make it as easy as possible. So if you don't reach that point yet, or you don't have the redo metadata loaded, or you don't see this object being created, just let us know. And then we can try to figure it out. And then from there, it should run smoothly when you're trying to run each lines. So the second step, what we need to do is on one side, we have all our fast results. So all the USI that were collected from the public domain to the one that we use to query. And the second one, the second thing that we did is to have the redo metadata that also contain the massive ID, so the same data set, and then the file name. And the idea is to merge those two together. So you have all the USI, now you want to attach any information that you can to those USI. So we need to merge those two tables together, and then we need to find the common identifier to do it. And the way we're transforming here, so we're creating a first part will be the function, two function that we create that modifies some of the column in those data frame to have the common identifier that we can at the end left join together. So the first step here is just to create a function. The second one is to transform the USI column inside the fast search data frame that all combined together. And the second function will do exactly the same on the redo metadata, and then you can left join those. So here we're gonna in the USI column, you should only have the massive ID and then the file name that are now merged together at the end of this portion. And here on this table, under the file name column, you're going to have some NAs because not all data set has redo metadata. So it's normal that you will observe some NA in this table, but other row will have different value to here. So now after you merge this, you should see this type of data frame.
I have a question.
Yes.
So, so the script I opened from the GitHub link that you just sent out is different from here. So where did you get this script? This is from the zip folder?
Yeah, it should not be different theory.
The script is called reverse metabolomics, right?
Yeah, I can.
Yeah, but there is one that is in Python and another one that is in R.
Oh, yeah, I downloaded Python.
So,
good catch,
oh,
Elaine.
probably.
Yeah.
Yeah, because the
Oh,
one in
yeah.
Python is in a Jupyter notebook format. I think I forgot the extension, but you should download the one that is .r in the end.
Okay. And so work direction, so we direct everything to the folder that we just created to put the CSV file or the folder, the zip folder that you just sent us.
So the working directory, so the first, when you choose a directory, should point toward the CMMC workshop folder. And the folder path that you have, that you have here, the folder path should direct to the input folder inside the CMMC workshop that we just set also in the chat.
Okay.
So here is just to basically when you have that folder path here, you can just hit enter tab after the forward or the backslash that you have here, and it will automatically you should see it like if you have the input.
The zip folder that we got is named workshop CMMC reverse metabolomics, and there's two subfolders here.
So you should have the input
the
folder,
input facet.
right? Yes.
Okay, I see. Okay,
yeah,
thanks.
Exactly. Input fast. And now all the CSV are inside that folder. So the folder path should direct to just the input fast, and then the code will automatically load the three data frames that are inside that input fast folder.
Okay, let me try. Thank you.
Nice. Let's continue here. So then we here what we do is just to standardize the table. So here we combine all the skin, the different part of the skin together, blood plasma and blood serum together with blood, and then we adjust for some of the nomenclature here just to have lowercase for all of them. If you are interested in studying a particular molecules that is found at the specific skin location, then you can comment that line and activate that line. Here, just to ease the analysis, we just combine all the skin together, but know that it's possible to inactivate that line to study a specific skin location. Same for blood. If you're interested in different plasma or different or the serum. And then what we want to do after we merge all that is to separate human data from rodents data. So here, what we want to do with those three small molecules that we just described, so phenylcholic acid, phenylene C4, and histidine C4, is to have their body part distribution and if they are more prevalent in specific diseases. So the first step that we do is we separate with only human data and only rodent data. So here we just filter our new data frame that have Homo sapiens 9606 for the human that we create. So this now data frame would only contains the human data. And here the next step is just to create a list of all the rodents that we have. And then we use that list to just create this new data frame that will only contains data about rodents. And then you should see one data frame like this here, where under the NCBI taxonomy column, you only have Homo sapiens data. So now let's continue. The other, now we're just going to explore these data. So we want to analyze, to create a function that's going to reveal the counts that we have for each organ in all the two different data frames that we just created. So here we're gonna visualize the data. So once you run those small lines, you should have, when you run the head body counts human or the head body counts rodent, then you should see this type of information showing up into your console. So here you should have the first sys row with different column, and then it will indicate the different counts associated with specific body parts and which molecules are found in associated with these specific body parts or organs or biofluid. Yeah, I think we have a lot of questions in the chat about the different code that are available on the GitHub account. So maybe it's the right time to just say it here. So we are today for today's workshop, we're showcasing the R script. Elena has created, she's leading the CMMC, and she has created the Python code, so script. So if you are more, if you know more about Python or Python is more helpful to you, we also have that script available in the GitHub. Here we're just showcasing the R environment because I'm just more comfortable with R. But know that we also have the Python script to please more users to do reverse metabolomics. So now after having all these this information about the body part, now we're trying to go and visualize that as a heat map. So we're just gonna rearrange this data frame to another format. So here we're just creating another function where we're gonna use the uberon body part name, which is the column that describe all the different body part organs or biofluid. And then you should have this type of data frame if you click on the pivot table humans or rodents. So now you're gonna have 4 columns, and each row will be different organs, and you have the number of count inside that table. And then, so just let me know if you're not there yet. But the next step is just to rearrange this table to make it in alphabetic order, and then place the column that is a character. So we have all the different body parts, blood, feces, jejunum, liver, and this column will go to the row name to make sure that it's compatible for visualization using our heat map. So here we're just gonna do what I just said. So I arrange the column around body part, and then we place that column to the row name. So now only our data frame will contain only numerical values. And this is why here we just make sure that it's only numerical value. You can define different colors to visualize your heat map if you don't like those colors by default here. You can log scale or not. So if you just want to look at the raw data, you can just not log the data frame. Here we're just going to log it for a better visualization. And then for human data, after running that last section of the code, now you should see the number of matches at the log2 of the number of matches. And then you should see that some specific metabolites are found in different body parts for human. And you're going to get the same. So for rodents, it's going to be just more matches because we can retrieve more body parts from rodents in general. So here you should see something that looks like this, where you have multiple matches across different body parts, organs, or biofluid. And then at the end, so the last two, so when you run this and generate these two heat maps, you have the option to export this as a PDF, and you can modify those settings if you don't like them, or just inactivate those lines if you don't want to import these heat maps all the time. So you just put a hashtag in front of these two lines. And they should be, when these heat maps are exported, they should be exported in your working directory, so in the folder that you're actually working in. So I don't see a lot of questions now in the chat, so I assume that most of you did get those heat maps, so I hope you did. Otherwise, feel free to ask questions. So then let's maybe continue. Oh, great, Celeste. Thank you. Nice. Did you use the R or the Python script?
I'm using R to follow along right
Great,
now.
great. Oh, that's awesome. That's awesome. Thank you. Great. So now the last part is to find if one of these molecules will be more prevalent in specific diseases. So here, again, what we do is we take the redo metadata, and then we're going to subset them, again, for human and rodents. Here, the code has more information because we wanted to provide you with a more thorough script to do answer different questions that you have. So not all of this is necessary for today, although all these lines should work smoothly. So here we're going to subset the redo metadata again, the redo for human data, and then for rodents data. And then we're going to filter for different information that are in the redo metadata. So we have a lot of information that is captured in the redo metadata. So I also encourage you to download the table or just open the table when you have some time to see what information might be useful to your own research in that table. And then by running all these lines, we're just going to kind of subset all the redo metadata based on the information that we are interested in. So for today, we're going to be focusing on the DUI common name, which has the disease information. So the next part of the code will be to actually target our two columns of interest that we're going to group. So we have our compound column that we're interested in and our disease DOID common name column that contains the disease information that we are interested in. And now we want to know the counts on how many scans were retrieved from those from the fast search. And then you reformat again the data frame for a better visualization, actually to be compatible for merging with the other tables. So here we have our group data frame that contains the information about the fast search, and then we have all the files that were present in the redo metadata, and now you should see a table like this. So the merged UID human data frame should have this information if you just click on it in your environment. So now you should have the disease information, the three different molecules that we search using the fast search interface, and then all the files that are currently available in Redo for um, all of these specific diseases. And the next step will be to normalize. So here we want to know if some specific diseases are associated with specific, if our molecules are associated with specific diseases. So here we want to take into account the number of files per disease that is currently available. So here, again, just run these lines, and then here it will just normalize everything according to the number of file that we have available in Redo. So now you should see a table like this, where everything is normalized. And then the next step is to actually calculate the percentage of this. So now we're just going to create the sum column-wise, and then multiply everything by 100 to get our percentage. And the last section of the code is the same that you already ran previously. So we want to remove the sum column that we just generated. We want to arrange our data frame by alphabetical order. And then we just want to place our character column as the row name to make sure our data frame only contains numerical values. And after this, you should see something that look like this after running these last lines here, where now you can see some molecules seems to be associated with specific diseases. So phenylcholic acid seems to be associated with some IBD bowel diseases, Crohn disease, or psoriatic colitis. And then we have RSNC4 that seems to be found in rheumatoid arthritis cohort. So hopefully most of you did get to that last heat map here.
Yes, it will. Excuse me.
Yes.
Yeah, this is nice. We were wondering whether the features in these disease data sets have already been filtered for significance, or is this plot just saying these are the data sets where you can find?
Yeah. So yeah, exactly. So this just show you association. So this doesn't do the statistical, you need to do it afterwards. It's part of the validation part. So here we're just trying to attach the formation that we have, the body part distribution, organ distribution, biofluid distribution, and then if it seems prevalent. Now in the validation step will depends on your questions and how you're going to validate these results, or if you want to do the statistical analysis. So what we suggest for the after you get these heat maps is to go back to the actual data set that these molecules are found in, and then proceed with the data processing, and then you can do your statistical analysis afterwards. But at least it will guide you to generate your hypothesis, or just have your question, formulate your question that you can address.
Yeah, thanks. It makes sense.
Yeah, so I think to follow up on this, so spectral count is crudely often associated with intensity, but it's not a perfect measure by any means. And as Vincent was saying, this is to help you formulate a hypothesis. You know, if your question is, does it reach a specific organ, or is it associated potentially with a certain disease, health condition? This allows you to get that first, be able to ask that first question. And then you have to actually go into the data, do peak area deconvolution. Hopefully there's enough metadata there where it says, okay, there's healthy controls, there's this health condition, or there's this organ versus that organ, that you will be able to do statistics on it. And that's exactly what we did in the papers, in the first 2 papers that are published.
Thank you.
Yeah, thanks, Peter.
Does anyone have any more questions about the script? All right, okay. The last thing that we want to showcase today is an introduction to our domain-specific masts, which again can help you answer the question, what is the origin of this molecule? So are these produced by specific microbes? Which microbes? Are they plant-derived? Which plants? Are they found in food? Which foods? So again, we have curated metadata for all of these. And today, we'll be focusing on micromast. Micromast, which we published a paper on it earlier this year, and we invite you to read that as well. It tells you how we got the metadata, the number of data sets we included, and we definitely have a lot of information on the species and strain level for this tool. And Vincent, if you want to go ahead and post the link for Micromast, please. Yes, we are recording the workshop. We plan to put it online, and we'll send out an email to everyone who attended as well. I'll let everyone get to the Micromast interface on GNPS. It's extremely similar to the FastMast interface that we were using earlier. And again, you can download a table with the mast hits, and the metadata is a little bit more comprehensive, more tailored for the species, strain, genus level for different microbes. And you can certainly adopt the script to make it more useful for this as well. And we invite you to, again, paste any one of the USIs that we used today. Vincent, if you want to paste the USI again, just in case anyone lost it. Thank you. This is the USI for phenylalanine cholic acid. The histidine C4 CSV provided is separated by a semicolon, so it's not parsed correctly when reading it in. So this seems like an issue with the naming of the file, and I think that the problem would be corrected if you renamed the file.
I think in this case, actually, I mean, for me, sometimes it happens as well if I open it like a CSV file in Excel, and then I don't know, I do like some kind of modification, and then I save it again. Instead of being a comma separated value, it will be semicolon separated value. I don't know why it happens. Maybe it was something that happened on that regard, Jerry, with you. Perhaps like if you just download the table and don't open it in Excel and just import it into the R script, it will probably work smoothly.
That's a good point, Helena. Thank you. Are there any more questions on the script before we look at Micromast? It happened to me when I cloned the project from Vincent's GitHub.
Yeah, I can replace those files on the GitHub if this is an issue. Let me send here the last table from the histidine so people can have it.
Thanks for letting us know, everyone. So if you re-download the file and rename it, I think the script should work.
Oh yeah, that is true. I actually checked here on Vincent's repository, and that one is indeed already deposited as semicolon separated. But well, that's good that we gave this workshop. Thank you for
Yeah.
letting us know. We will fix that.
That's awesome. Yeah, again, this is our first workshop on this, so thank you all for joining and helping us work out some kinks. But if you hit the search micromass by USI, if you're following along with this step, and you scroll down, again, you'll see a table that you can export and play around with, see the matches, same information, delta mass, cosine score. And you also get an interactive tree, which will show you the levels of the information on each microbe that we have available for the matches. And we even have a lot of strain level information, which is awesome. And please feel free to play around with this tree. If you hover over it, it will show you how many data sets and how many matches there are for each bubble. And a lot of this has been manually curated to include a lot more information on the phenotypes of each data set. And you can absolutely adopt the script to include this information and make your own heat maps, let's say for you want to know the species level for a certain metabolite. And we definitely invite you to do that or ask us any questions if you have on that. But really, I think that we've given you guys an introduction to the tools and worked you guys through the script. Wow, so interesting. Yeah, the trees are awesome. What's the difference between micromast and microbiome mast? Yeah, I think that's
Yeah,
a
I can
great
answer.
question for Vincent.
Yeah. So we are currently developing microbiome mast. So the microbe mass contains monoculture information. So only monoculture information. And we have limited information about the community, how they're growing together. So it's something that we're currently developing to try to include or develop our new platform where we're going to have community growing together and probably reveal more function of the microbes. As we know, most microbes are not living by themselves. They're surrounded by a lot and thousands and hundreds of different microbes, and this is what we're trying to grasp here. So that will be one of the differences in the microbiome mast that we're currently building.
Yeah, that's a great question. So maybe just to recap some of the tools that we went over, part of GNPS in our lab is the GNPS libraries, where we got the MS spectrum to import into our FastMass tool on GNPS, which searches all the repositories for that spectrum. We also showed you guys Micromast, which specifically only looks in data sets which have associated metadata for microbial monocultures. And we walked you guys through the script, which can begin to make hypotheses about the sort of associations that each metabolite can make for organ or tissue type, disease state, And if you look more into the script, you can really filter that table for anything. And kind of it's a choose your own adventure, I would say. And
I want to follow up on the question of masks versus micromasks. Conceptually, they're essentially the same thing. In fact, what we do, even in micromasks, we search all the public data and then just filter for microbial data. The difference is, so if you look at masks, the metadata that's connected there. So we search currently about 1 million LCMS files that are across the 3 repositories. About 700,000 of those have some sort of metadata. So a lot of public data doesn't really have metadata. We spent some time trying to harmonize this metadata. So this is a postdoc, Yasin El-Biad, that did the harmonization. And the type of things that we could really easily harmonize were disease states, organ distributions, and similar type of things. But there was a lot of things that was a bit more difficult to harmonize. And one of those was actually microbial taxa. And so there was a lot of data in the public domain of microbes grown in culture that didn't have any metadata. And so we put a call out to the community, hey, if you have data in any of these three repositories, let us know, and we would like you to curate each of the files with respect to taxonomy, that is NCBI taxonomy, so that when you then search with an MSMS vector, you get taxonomic information. The micromass is really focused, and we created a very specific interface for micromass that you can put in that entry and actually explore the tree itself. Whereas if you do it via mass, you would have to download the data table, do the data science, and build trees or whatever you like. And so that's really the difference, where we serve specific goals basically in these domain specific mass. So we have plant mass now, we have food mass. So if you care about food metabolites and potentially what foods they're distributed in, plants is taxonomy of plants, microbes, taxonomy of microbes. Microbial mass will be expanded to do 2 things. One, that you can go from the gene side and learn a little bit more about what molecules may be co-occurring with specific genes. But that will take a little bit longer to build. But also anything you care about in terms of microbiome analysis. Does your specific molecule change in the context of diet? Does it correlate to health conditions? Does it are communities able to make them? So a lot of people have these communities that they're culturing, and so they will have different pictures. And so there we're going to go much broader, where you can then learn as much as you can about microbial molecules and how it's distributed. But we're also creating drug mass, so focus on medication specifically. And then the other one that we're creating is sort of exposure mass. So depending on we're all exposed, or plants or aquatic ecosystems, we're all exposed to different things that humans make. And so personal care products, insecticide, pesticides, drugs would be also part of this. So it makes it easy to track where that information is. And so these domain-specific mass really serve specific communities, whereas the mass itself is sort of the broad scale, looking at everything. Hopefully that helps a little bit more.
Thanks, Peter. I think that was a great explanation of all the different tools that we have available and that we want to build. One thing that we also want to make available, because this is an online Zoom workshop, so you're definitely going to have more questions and maybe want to spend more time on these certain parts of the workshop that we showed you. So we are offering 3 office hours. We plan to do 1 to 2 hours. The first one will be this Thursday, same time, 9 a.m. Pacific Standard. And the Zoom link, I'll send the Zoom link out here. And we hope that not only you have questions about the tools that we showed today, but maybe that you'll try this with your own metabolites and have your own data, and maybe have questions on how to further explore, and maybe questions on step 4, the validation. So that would be like the question that we got asked earlier about, is this showing statistical analysis of each feature? And we can certainly show you guys the right direction to go, and in the ways that maybe we've used that in some of the previous publications. And of course, for any updates on the CMMC, we do have Blue Sky. And if you have any more questions for I or Vincent, who developed this entire script and the nature protocol, please feel free to reach out to any of our emails. Vincent, if you have any final words.
Yeah, no, this is great. I hope you enjoyed this workshop today. Feel free to reach out at any point if you have any question through email, and hopefully we'll see you guys, some of you on Thursday, to try to go through your own molecules. So yeah, please feel free to reach out if you have any question. I'll be glad to answer and talk with you about your research.